Protein structures dictate all biological functions. Knowing their exact three-dimensional shapes lets us see how they latch onto nutrients or signal a cell to divide. Yet, physically determining those shapes has historically been the most significant and time-consuming bottleneck in scientific research. DeepMind's AlphaFold changed that by training a deep learning model to predict these structures directly from amino acid sequence data in a matter of hours. If a computer can predict a structure this quickly, does it mean researchers can safely abandon millions of dollars of physical lab equipment? Because a digital prediction can miss the physical nuances of a living system, researchers must weigh the speed of the algorithm against the specific operational trade-offs of the lab. The gap between physical lab techniques and modern algorithm-driven output comes down to labor and speed. This compares historical baselines between these two methods. The left column shows traditional methods, which demand significant capital and years of trial and error to isolate a single structure. The right column shows how AI flips this, offering insights in an afternoon at almost zero marginal cost. This disparity shifts the dilemma for lab directors. The question is no longer what can we afford, but rather what level of structural certainty do we actually need? Traditional structural biology relies on an experimental triad: X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy. The primary input for these methods requires grueling physical sample creation, such as forcing proteins to form crystals or flash freezing them in a vacuum. That labor investment buys you one critical output: empirical, atomic-level proof of a specific biological structure. Physical lab experiments are uniquely able to capture these proteins while they are actively ratcheting and shifting between different functional shapes. Lab methods limit your iteration speed, but their empirical certainty is necessary when studying complex molecular dynamics. Before AlphaFold, researchers used legacy computational methods, like homology modeling, as a cheaper fallback to skip physical lab work. The utility of these models stops the moment you sequence a protein that doesn't look like anything already in the database. AlphaFold bypassed this by training its neural network on the entire protein databank, allowing it to predict structures from scratch. However, as the complexity of the system increases, the prediction quality drops. The AI struggles to model how multiple proteins interact inside large complexes. AlphaFold provides a highly educated static prediction, but without dynamic, empirical proof, it cannot yet fully replace a physical lab. Selecting a strategy requires mapping these tools directly against an institution's budget and the necessary resolution. Plotting these tools on a quadrant chart reveals their trade-offs. Traditional experimental methods sit top right: high cost but unmatched empirical dynamic resolution. AlphaFold lands bottom right: low cost and high static accuracy, but low dynamic data. Legacy software sits bottom left: low cost, low overall accuracy. This matrix shows that there is no universal best tool. Researchers must choose based strictly on their most pressing operational constraint. Moving from theory to practical application means labs must filter their strategy through a specific decision-making process. This decision tree shows the primary filter. You must decide, does the goal require raw speed or empirical mechanics? For speed, the branch leads to AlphaFold. For empirical detail, it routes toward traditional lab methods like cryo-EM. Categorizing the work this way clarifies whether a lab's budget should go toward cloud computing or specialized hardware. Small academic labs use the AlphaFold route to democratize their research, allowing them to compete with larger institutions. Immediate structural insights prevent these teams from spending months of funding on lab assays that may never yield a result. Large pharmaceutical companies use a very similar strategy to rapidly sort through tens of thousands of potential molecular targets. Pharma uses AI to screen more candidates in a week than a physical lab could test in a year. However, the speed of AI is only a starting line. A predicted model must still pass empirical verification before human drug trials begin. Following the empirical branch, structural biologists still rely on X-ray and cryo-EM to observe actual molecular machines at work. Evolutionary biologists use the AI path to predict structures for proteins that have no known historical precedents to study. This has created a hybrid gold standard workflow. Labs use AlphaFold's rapid predictions to narrow the focus of their expensive physical experiments. These methods are no longer competitors. The AI generates a hypothesis, and the physical lab proves it. AlphaFold provides a high-resolution map of protein possibilities, allowing researchers to save their traditional lab tools for the hardest ground.